Welcome. It is my great pleasure to welcome you to the New Jersey Academy of Science inaugural Advancement of Science Awards seminar. Today, we will be recognizing Dr. Deepak Sarkar for his multiple seminal discoveries on the impact of excessive alcohol on endocrine and neuroendocrine regulation of cell function and metabolism. He identified the role of hypothalamic neurohormones in the regulation of reproductive activity, stress response, immune function, and cancer. Dr. Sarkar's work leading to these outstanding discoveries deserves to be recognized by the New Jersey Academy of Science. We are very grateful to the seminar organizing committee, particularly Dr. Abby Sarkar, Ms. Sophie Sarkar, and Ms. Stacy Pontotiero, for putting together an engaging program today. I would like to take this opportunity to inform you about the New Jersey Academy of Science many initiatives that have taken place in the last couple of months. This past July, the New Jersey Academy of Science board announced the winners to the virtual research competition. I would like to extend my congratulations to the winners of the Junior and Senior Academy and the New Jersey Academy of Science judges who helped us despite the current health crisis. In addition, the seminar called A Closeout with COVID-19 Heroes was a great success. There was 110 participants registered, and presently the video is available for streaming and already has received 70 plus likes. Please support the New Jersey Academy of Science and follow us via YouTube. All information is in our website. I would also like to notify our members that July 2020 was the month that we got the most donations, and I hope our members in the New Jersey community will continue to support the New Jersey Academy of Science. Please, let me inform you that the Academy is 100% funded by its membership, publications, fundraisers, and private donations. It is our hope that our New Jersey Academy of Science members and friends will continue to contribute to our mission. In the months to come, we will continue to sponsor more seminars regarding and targeting the Junior and Senior Academy members. For example, some uncommon titles include Cybersecurity and Privacy, Environmental Pollutants and Human Health, and Career Connections, BSMD programs. More specific information, including seminar dates, will be posted soon. I sincerely hope you will enjoy today's seminar. Thank you for your participation. At this moment, I would like to introduce Dr. Abby Sarkar, who will be our host for the seminar. Dr. Abby Sarkar is a postdoc in Rola Nusset's lab in the Department of Developmental Biology at Stanford University. She completed her PhD at Harvard University. She's currently studying the cellular and genetic mechanism of liver cancer with the hope of identifying new biomarkers and drug targets for this often fatal disease. Dr. Abby Sarkar will be introducing the speakers for today. Again, I would like to extend a very warm welcome to each and every one of you. As always, should you have any questions, suggestions, clarifications, or just want to say hi, do not hesitate to post them in the chat mode. Thank you very much for listening, and let us welcome Dr. Abby Sarkar.
Hello, everyone. My name is Abby Sarkar, and I'm calling from the ancestral and indigenous lands of the Ramaytush Ohlone people, now called Palo Alto, California, Stanford University. First of all, I want to thank the New Jersey Academy of Sciences for the opportunity to co-host this program of exciting speakers, as well as an award for my very first and ever-inspiring science mentor, Dr. Deepak Sarkar, who happens to be my father. I'm also very grateful that this is an open and inclusive meeting. Everyone who wanted to could sign up for this meeting for free. And finally, I want to thank all of you for taking time out of your Saturday to join us during this unprecedented and difficult time. I hope this seminar can give a moment of light. We have a great list of speakers today. There will be 2 talks followed by a short break, another 2 talks, another break, and then finally the award ceremony. During the talks, please feel free to write comments and questions in the chat box. We will address the questions during the Q&A session after each speaker. Talks. I'm happy to introduce our first speaker, Dr. Ryan Logan, who is an assistant professor at the University of Pittsburgh School of Medicine. He specializes in circadian biology and drug abuse, and his talk today will be on novel sleep and circadian mechanisms linking rodent and humans in opioid addiction.
Thank you, Dr. Abby Sarkar, and also thank you to Dr. Agapito for organizing a great afternoon to honor Dr. Sarkar. And I'm honored to be amongst the speakers today to share some of the newer stuff that we've been doing in the lab. So can everybody, Maria, can you see this?
Yes.
Perfect. So the title of my talk is going to be Novel Sleep and Circadian Mechanisms Linking Rodents and Humans in Opioid Addiction. And I'm just going to highlight one of the things that we've been doing to look at how circadian mechanisms in humans might contribute to opioid dependence, and the parallel animal studies that we've been conducting to actually look at how these signaling mechanisms contribute to opioid addiction. So I just have to give a conflict of interest disclosure. Consultant for Pfizer, and I've received funding that doesn't impact this science from a pharmaceutical company. Just a brief background of my training. So I went to University of Maine. Most of that research focused on circadian rhythms and addiction, and I had a few postdoc choices. And one of those postdoc choices was actually in the lab of Dr. Deepak Sarkar. And I remember showing up to the interview, and after a long day, Dr. Sarkar turns to me and he says, "So are you ready to give your talk?" And I didn't have a talk. So I basically gave a talk on a whiteboard. Well, I must have done okay, because when he drove me back to the train station in New Brunswick, New Jersey, he offered me the position, and thankfully he did. He continues to support me. I talk to Deepak fairly regularly. I now see him at meetings all of the time, and he's always interested in how things are going, both scientifically and personally. After Rutgers, I went to the Jackson Laboratory and then found myself at Pitt, where I opened my own lab. Our lab is mostly focused on circadian rhythms in addiction, and we use translational models to understand something about human disease. The models that we primarily use are rodents, both rats and mice. And we have a number of different avenues of research in the lab where we tried to kind of bridge human disease to rodent models, both forward and reverse translation. And these are just kind of examples of things the lab has published in and continues to do. And what I'm going to highlight today is some of the work that we've done in human postmortem brain tissue and how we can bring molecular insights from the human brain into our animal models to understand something about the mechanisms of addiction. Our lab over the past 2 to 3 years has become extremely invested in the opioid crisis. And this goes without saying to most of this group, but we're really in a troubling time with battling opioid overdose deaths, and also increases in diagnoses of opioid use disorder. And so over the past 5 years, what's actually become apparent is that the main opioid that's being abused has transitioned from heroin to highly potent, very powerful synthetic opioids. And this has driven a large surge in both misuse of prescription opioids, but also people that are using prescription opioids or synthetic opioids, and also transitioning to a clinical diagnosis of opioid use disorder. One of the hallmarks of opioid use disorder, and this is kind of emerging over the recent decade, is that sleep is completely disturbed, both during when somebody is using opioids, but also withdrawing from opioids. And this is just a statement from NIDA several years ago, saying that sleep deficiency, including insufficient sleep duration, irregular circadian rhythms, and poor sleep quality, is highly prevalent. And actually, the clinical data that's coming out over this past 12 months has shown that almost 95% of individuals experience some clinically diagnosable circadian and or sleep disorder that's comorbid with opioid use disorder. This is just a quote from an addiction psychiatrist that I work with closely in our mental health facility here, Western Psychiatric Hospital. And one of the things that she commented on was that during clinic, one of the most common complaints I receive from patients being treated for opioid dependence relates to poor sleep, heightened arousal, and agitation. And one of the things that struck her was that many patients, when asked why they relapsed or what was one of the contributing factors for why they relapsed, was to acutely relieve these burdens. And so we think that this could be a major contributing pathway to both opioid dependence and opioid relapse. So our lab kind of works from this conceptual model of how sleep and circadian rhythms drive different neural circuits in the brain. And when you disrupt sleep and circadian rhythms, you impact neural circuit function that ultimately has consequences on things that we think contribute to vulnerability to addiction. And so I'm going to talk about one of those studies that we're doing in this conceptual model. And we know also that both drugs of abuse, but also addiction itself, can disrupt sleep and circadian rhythms. And so there's this bidirectional interplay between sleep and circadian rhythms that contributes to addiction vulnerability, but also chronic drug use and drug dependence can feed back and disrupt sleep and circadian rhythms. I'm going to go over this fairly quickly, but I just wanted to give you highlight a few key points of things that I'm going to talk about. We do a lot of work with the circadian timing system, both across the entire body, but also in individual cells. And across the entire body, the light is the biggest entrainer to circadian rhythms or clocks in the brain, but also throughout every major organ in the human body. Almost every cell in the body expresses what's called the molecular clock. And these molecular clocks are a series of positive and negative feedback loops that control rhythmic function in different tissues and cell types. And one thing that we've been realizing, especially over the past decade and a half, is that the things these clocks actually control differ significantly between tissue and cell types. And so we're very much interested in how these clocks in individual cells that obviously make up an organ contribute to both health, homeostasis, but when disrupted, contribute to disease processes. So a lot of our work has been done in animal models looking at what these molecular clocks are doing. But several years ago, we actually designed an approach that stemmed from a paper in PNAS from Huda Akil's group and William Bunny's group, showing that you could actually recreate these molecular clocks or these molecular rhythms in the human brain from subjects who deceased. And we created an algorithm where we used the time of death of the subject to order people around a clock in relation to sunrise on the date they died, but also considering the place they died. When you do this, and your sample size is large enough, you can actually look at subjects across the entire day, and you can recreate some of these transcriptional rhythms in the human brain. For this particular study, we focused on two key nodes of the addiction circuitry, the nucleus accumbens and the prefrontal cortex. Now, the only thing that you really need to remember is that the prefrontal cortex and nucleus accumbens have bidirectional synaptic projections, okay? And the prefrontal cortex seems to be a major substrate of decision-making, critical for regulating impulsivity, while the nucleus accumbens is a major neural substrate of reward, drug reward, salience, and motivational value. And these two brain regions, amongst others, form a circuit. And this circuit seems to be a critical player in both the hallmarks of addiction, such as cognitive disruption, poor decision-making, and increased impulsivity, but also the maintenance of addiction or the transition to dependence. So we use these two brain regions in human postmortem brain. We have a fantastic brain bank at the University of Pittsburgh. And we isolated the nucleus accumbens, which is part of the ventral striatum, and the dorsal prefrontal cortex, which is part of the frontal lobe. And we did 20 subjects of unaffected controls compared to 20 subjects with an established diagnosis of opioid use disorder who were also actively abusing opioids amongst other drugs. And we just had one simple question. Let's go on a fishing expedition. So we did bulk RNA sequencing on these two brain regions. And then we used our time of death analysis to determine whether transcriptional rhythms in the opioid use disorder brain were disrupted. And if they are, should we even care? So this is just a heat map of the top 200 genes in the nucleus accumbens in our control subjects versus our OUD subjects. Zeitgeber time is just what we standardize time of death to. So Zeitgeber time 0, ZT0, is actually sunrise, normalized to each subject's date of death. And as you can see, what you'd expect if there were transcriptional rhythms in the control brain, we would see these waves of yellow and these waves of blue that vary across time of day. And that's what we see. When we look in the opioid use disorder brain, these rhythms seem to be entirely disrupted. This is just an example of two genes and the method that we're using. So each one of these dots is a subject. On the y-axis is expression value. On the x-axis is, again, Zeitgeber time. And you can see for CART, which is a known circadian gene, and OPRL1, which is a nociceptin gene involved in opioid-induced analgesia and also pain, is highly rhythmic in the controls in the nucleus accumbens and loses rhythmicity in the opioid use disorder brain. So these are just two examples of hundreds of genes that we found to be arrhythmic in the opioid use disorder brain. So we wanted to get something functional out of this data. So can we leverage this data to gain any insight into mechanisms that we can actually test? So we started to group changes in the molecular rhythms in the postmortem brain of OUD subjects by whether the rhythm was lost or whether the rhythm was gained, okay? So if they were lost in OUD, they were present in controls. If they were rhythmic in OUD, they were not rhythmic in controls, but become rhythmic in OUD, and that's gained. And as you can see, when we compare the number of genes that are rhythmic in controls versus the number of genes rhythmic in OUD, there's very, very little overlap, suggesting that a gain in rhythms in OUD might also be involved in the pathophysiology of OUD, in addition to what's lost or what becomes arrhythmic in OUD. So we did a pathway analysis using the information of what's lost of molecular rhythms in the opioid use disorder brain in the accumbens. And we just asked a very simple question. Of all the genes that are changed, what are the top transcriptional regulators that are predicted to regulate these transcripts? And the top three, HNF4A, BMAL1, and MPAS2, are all known circadian transcription factors. One of which, NPAS2, is something we were already studying in the lab. So serendipitously, we found something that we had found in the mouse and now also in the human. And so we exploited this a bit. We took male and female mice. We treated them with chronic opioid administration using fentanyl, IP injected over 14 days. And then we just looked at whether MPAS2 gene expression was changed in the nucleus accumbens of male and female mice. And voila, we see a significant reduction of MPAS2 in the nucleus accumbens when those mice are exposed to chronic fentanyl. So now we have a reduction in the human nucleus accumbens that's associated with opioid dependence. And we also now have a reduction in expression of MPAS2 in the nucleus accumbens when mice are exposed to chronic fentanyl. So we designed some mice using CRISPR-Cas system, where we basically deleted or genome edited out the basic helix loop helix domain of the MPAS2 protein. Now, this domain is particularly important for MPAS2's ability to bind a coactivator called BMAL1 and regulate clock-controlled gene transcription. So the hypothesis was, if we delete this basic helix loop helix domain, it can no longer bind its coactivator, and you lose transcriptional rhythms in the accumbens by a deletion of MPAS2 function. So we did this, and we developed a model where we can tease apart different components of drug-seeking behavior. We developed an operant oral self-administration model of fentanyl in mice. We also have an intravenous drug self-administration model working, but we wanted to use oral self-administration because we realized, particularly from the newer data that's coming out in epidemiology, that a lot of people are actually abusing opioids orally, mainly because these synthetic opioids like fentanyl, like carfentanil, like remifentanil, are highly potent, and you can reach the same level of euphoria or high without having to intravenously injecting through a needle. And so we can tease apart different components of addiction using these models. We can look at how fast an animal actually acquires the drug-taking behavior, how motivated they are to take the drug, and whether they relapse when exposed to cues that were previously associated with a drug presentation or drug taking. And so we took our MPAS2 deficient mice, we took males and females, we put them through this oral self-administration, operant oral self-administration model, and we asked, how hard will you work to consume an oral containing solution of fentanyl? And so in this top panel are males, and this bottom panel is females. And panel A is just reward number given. So how many times they press the lever to disperse this fentanyl-containing solution over the number of sessions on the x-axis. And then we can also measure from the solution that they dispersed how much they actually consume of the drug. And what you can see is in males, opposed to females, is that these MPAS2 deficient mice actually show an increased acquisition of fentanyl oral self-administration behavior. Females overall, even though there's no genotype effect, females overall, compared to males, tend to self-administer more fentanyl. And so right now we're playing with different doses and different training paradigms to see whether we can tease out the genotype, the sex, and the sex by genotype effects. When you look at how motivated they are to respond to fentanyl-containing solution in males and females, again, male MPAS2 deficient mice actually will press the lever a lot more across multiple doses for fentanyl before they give up, okay? So they've increased motivation to consume the fentanyl-containing solution. When you try to extinguish that behavior over a number of days, MPAS2 mice have a very, very difficult time extinguishing this behavior. But when you present a cue or a combination of cues that were previously associated with the administration of fentanyl, they actually respond more. And so we can actually measure, using this oral self-administration paradigm in mice, both the acquisition, drug-seeking motivation, but also the propensity to relapse. And again, MPAS2 mice, male mice, seem to do this more overall compared to females. So we identified MPAS2 in the nucleus accumbens in both human and mouse. And we know a lot about the circuitry or the microcircuitry in the nucleus accumbens. And there's really two main cell types that I'm going to focus on briefly. DRD1A expressing, these are supposed to be reversed. DRD1A expressing, or called D1 positive, versus DRD2 expressing, called D2 positive. And for all intensive purposes, there are a number of caveats to this, but for all intensive purposes, D1 MSN activation actually promotes drug-related behaviors, and D2 neuronal activation in the accumbens actually antagonizes drug-related behaviors. So we had an initial hypothesis that maybe MPAS2, because when you delete it, is driving, when you delete it in the accumbens, it's driving fentanyl self-administration, that maybe MPAS2 is doing something in D1 neurons to promote drug reward and seeking behavior. So we used an approach called RNA-scope, where we basically used different probes that target different transcripts in the mouse brain. And we targeted D1 expressing neurons in the nucleus accumbens, D2 expressing neurons in the nucleus accumbens, and also the MPAS2 gene. And we mapped the entire mouse striatum using RNA-scope, and we just quantified how many D1 neurons in the nucleus accumbens versus how many D2 neurons in the nucleus accumbens express MPAS2, with the hypothesis that if MPAS2 is doing something to drive reward-related behavior, maybe it's doing so in D1 neurons. And that's what we saw. We saw about a twofold enrichment of the number of cells expressing MPAS2 in D1 neurons than what we saw in D2 neurons. So again, we went back to our CRISPR toolbox and we designed a particular intersectional genetic approach where we could delete MPAS2 specifically in D1 neurons in the ventral striatum using multiple genetic drivers. And we used a different paradigm to gather some pilot data to see if this was going to work. This paradigm is called drug condition place preference. So in its simplest form, a mouse is put into a 3-compartment box. Over a series of days, the mouse is administered a drug in one of 2 of these compartments. And then on the test day, you put the mouse in the box and you say, hey mouse, explore this box and let me know how much time, or let me know which side you prefer based on the amount of time you spend on either one of these compartments. Now, mice who have been exposed to a number of drugs of abuse, including fentanyl and morphine, tend to spend more time on the side for which they received the repeated drug administration. So we gave fentanyl at a fairly low dose, 0.1 mg per kg in CPP, and we saw what we expected to see. So if you delete MPAS2 in D1 neurons, and I'm only showing the male data, versus D2 neurons in the accumbens, you actually increase CPP score, meaning MPAS2 deficiency in D1 neurons in the accumbens drives condition reward for fentanyl. And so this is consistent with what we saw for the NAC-specific MPAS2 deficiency in driving drug-seeking behavior during self-administration. So we wanted to connect this to sleep. Now in humans, opioid dependence is accompanied by marked changes in sleep, particularly reduced rapid eye movement sleep, REMS. And we think that this reduction in quality of sleep, particularly in this reduction of REM sleep, is thought to contribute to craving and relapse. And so we took our MPAS2 NAC specific deficient mice, and we looked at REM sleep throughout over 7 days and quantified the amount of REM sleep spent during the light and dark phase. And importantly, what we saw this fairly massive reduction in REM sleep in MPAS2 deficient animals compared to wild types. And so right now, what we're trying to do is trying to figure out whether MPAS2 in the nucleus accumbens could be a molecular switch that drives different drug consequences. Okay, so under normal conditions, MPAS2 binds its coactivator BMAL1, you get beautiful transcriptional rhythms. We know that these transcriptional rhythms contribute to homeostasis within the microcircuit and then healthy behavior, okay? So under normal conditions, MPAS2 does what it needs to do. Under opioid conditions, MPAS2 actually, and I didn't show you this data because I didn't have time, but MPAS2 actually loses its normal binding partner, binds something else. We're trying to figure out what that coactivator is. And you actually lose or gain transcriptional rhythms. MPAS2-driven rhythms are lost. These loss in transcriptional rhythms in nucleus accumbens contribute to drug-induced synaptic plasticity, potentially sleep and arousal changes associated with chronic opioid administration, and contribute to drug craving and relapse. So this is the working model that we've been focused on recently. So with that, again, I want to thank the organizers for this afternoon. I want to thank for the invite, truly honored. It's nice to see everybody. It's nice to see familiar faces. I want to thank my funding. Most of this funding has come through recently on some of these newer studies. And I want to a special thank you to the families who donated tissue to our brain bank at extremely difficult times. I also want to thank collaborator Marianne Sine, and Jill Glossier and David Lewis, who have been helping me with the human postmortem studies. And of course, my lab, who has been learning how to deal with me during COVID-19. So thank you.
All right, thank you, Dr. Logan. We're going to open up the Q&A session now. If anyone has a question, please write it in the chat box. There is a question from Xingping Pang, who asks, does NPAS expression level differ between male and female mice? And does NPAS expression, is it dependent on neuronal activities?
Yes, so we have not yet seen sex differences in MPAS2 expression. We're currently working that out right now. It's a possibility that there are cell type specific expression differences of MPAS2 between males and females. MPAS2 is not like the other MPAS genes. So recently there's been a lot of play with MPAS1 and MPAS4. Both of those genes are immediate early genes. MPAS2 is not an immediate early gene. And so it's probably driven by changes in overall activity within the neuron. It's expressed. We have some data to suggest that. But it seems to be more importantly what it's doing downstream to its transcriptional targets that is mediating the effects of drugs of abuse on these cells in the nucleus accumbens.
All right, and Kunal Sikdar is asking what NPAS2 knockout is it a gremlin one, and does it affect growth and development of the brain?
Yes. So I took some slides out because I didn't want to bog people down with the boring genetics of how we created these mice. We actually created these mice through an intersectional genetic approach where we split the Cree construct into two separate lines. And when the Cree construct recombines, when you cross those two lines, it's inducible. So we can now specifically control NPAS2 overexpression and knockdown in an inducible manner. So this is not a germline knockout that would have secondary effects on overall development.
Okay, not germline. Sorry for the error there. I have one last question. What do you think about opioid abuse is reducing the NPAS2 expression? Is it an epigenetic mechanism involving methylation of the promoter?
Sure.
Is that why even after use of opioids and remission, there's still the circadian effect?
Yeah, so there's kind of two answers to that question. The first part of that answer is we're testing whether NPAS2 is first a critical driver in these opioid-induced effects through NPAS2 rescue and overexpression. Okay. Once we have that, then we'll go into basically what you are suggesting are opioids or whatever opioids are doing in the nucleus accumbens epigenetically modifying the gene or the protein activity. We have some data in collaboration with the Yale NIDA Proteomics Core to suggest that it might actually be a post-translational modification of NPAS2 that could be driving these effects. However, actually I just submitted the paper this morning, is that we have some insight into how this gene is epigenetic regulated from our human postmortem data. And what we saw with the human postmortem data genetic locus and also loci surrounding the NPAS2 gene, particularly in the promoter, in unaffected controls, it seems like a fairly stable epigenetic state, meaning there's some transcriptional activation. It seems to be driving kind of low stochastic states of expression. But in the OUD brain, we actually see epigenetic marks at that locus that are associated with transcriptional repression, which is what you'd expect based on our data. I don't think it's promoter methylation. I think it's a secondary histone modification. Yeah.
One final question. HIF1 alpha, beta, ARNT expression is elevated in response to coping with neuronal O2 deficits. Is it all involved in OUD, especially in P2C4 neuronal loci?
Yeah, so I could talk for an hour about this, but it's entirely possible. So if you look at enriched pathways in the OUD brain, and I didn't present this data, but if you look at enrichment of pathways in the OUD brain compared to unaffected controls, one of the top pathways is hypoxia, okay? And so we have kind of a working model that there's a possibility that both hypoxia, neuroinflammation are contributing or at least feeding in to these pathways that regulate circadian rhythms in the nucleus accumbens. So yes, it could be associated with neuronal O2 deficits. Yeah.
Great, thank you so much. All right, we're going to move on to the next speaker. Dr. Sushanto Banerjee, who's a professor at the University of Kansas Medical Center. He has had a long career in cancer biology, and his research focuses on cancer endocrinology and therapeutics. His talk is titled Stimulating CCN5 to Target Triple-Negative Breast Cancer. Dr. Banerjee, if you could share your screen.
So, okay, let me start. Sorry for delay. Okay. So today's, can you hear me now? It's okay? Everybody can hear?
Yep, you sound
Yes.
great.
Okay, great. So, okay, the first of all, it is a great honor and a privilege to be invited to give a talk in this great occasion. And just a minute, my computer is, okay, go ahead, okay. I'm sorry, I thought time differences make me. And I'm delighted to be here today. I wish to thank Dr. Agapito, President of the NJAS Academy of Science, Kent University, and Dr. A.B. Sharkar for his very kind invitation. I know A.B. when she was 1 year old, I think. Now she's a lady. I bring greetings and best regards and wishes from my lab members and my family members on this NJAS inaugural Advancement of Science Award celebration. And finally, my special congratulations to Dr. Deepak Sharkar for this award. Okay, you know, there is no such thing as a self-made man. So I am very sure Dr. Sharkar is not a self-made man like other people, but he is a famous scientist. We will reach our goals only with the help of others. So my special thanks goes to his family members, including AB, Sophia, both are here, because without them he could not be here. It is same to me and others also, and thus I want to acknowledge my different faculties, the members, and also my universities. I am from the Kansas City VA Medical Center, and also from University of Kansas Medical Center, University of Kansas, yes, and Department of Pathology. Before I start my talk, I have to acknowledge those members who are involved all my work in this. So, oh my gosh, this is not working. I don't know why. Okay, this is my VA Medical Center. You can see. Okay, okay. Let me fix my PowerPoint. It is really. Okay. Sorry, sir. Give me a time. Okay. So welcome to the Kansas Biomedical Medical Centers, and then you can see my university. I'm attached to the School of Medicines, and I want to acknowledge those people who are involved in this work. That is, I don't want to go details, but mainly I want to acknowledge my wife Snigdha Banerjee. She is involved in all kinds of work, handling my labs, and also in Amul Haq, Gargi Maiti, and my special thanks to Dr. Douglas McGregor, who was my pathologist, and I learned the pathology from him, and he's my guru, one sense, okay, and other people's also. So, as you know, the breast cancer is the most common cancer among American women, except for the skin cancer, and it is now applicable globally. About 1 in 8, which is about 12% women in US will develop invasive breast cancer during their lifetime. Breast cancer, if you look at the sequence of the breast cancer development, it is basically like all other cancer. It is a sequential disease initiated with a hyperplasia, which is called overproduction of the cells, atypical ductal hyperplasia, ductal carcinoma in situ, and invasive cancer. These are basically based on their pathology. There are two types. One is atypical lobular hyperplasia. This is a ductal carcinoma and lobular type. Based on this pathology also, the pathology is divided into two groups. One is called pre-neoplastic groups, other is neoplastic. This pre-neoplastic, within the pre-neoplastic conditions, hyperplasia, atypical ductal hyperplasia or ductal carcinoma in situ is considered pre-neoplastic disease, while invasive ductal carcinoma and considered as a neoplastic disease. This is a very long process. As you know, it will take from normal to invasive ductal carcinoma and even ductal carcinoma or even ductal carcinoma in cells to DCIS take almost 15 to 20 years. So it is a very, not a slow process, it's a long process. Start from maybe when you recognize the disease, it started 10 or 15 years ago. So if you look at the histology of the disease, you can easily recognize the disease. This is called ductal carcinoma in situ, and these two is invasive ductal carcinoma. It is a differentiated type and it is a non-differentiated type. If you look at the DCIS, how to go back? If you look at particularly the DCIS type, you can see two different types of cell layer exist. One is called the myoepithelial layer and other is called the epithelial layer. Myoepithelial cells plays a vital role in the progression of the disease. These myoepithelial cells, that's why it is called line of control. It's control the whole these systems. So if you destroy the myoepithelial cells, the disease immediately start to progress. I will show some data in evidence of that. Once the myoepithelial cells disappears, the epithelial carcinoma cells disperse throughout the breast, and eventually it moves to different organs. It's called metastasis. Next slide will show the algorithm of the breast cancer type. All breast cancers are basically three different types. One is progesterone or receptor type, the progesterone receptor positives or estrogen receptor positive, which is about 65 to 75% of the disease. Other is the HER2 positive, which is 15 to 20%. And third one, the triple negative breast cancer. So this progesterone or estrogen receptor positive breast cancer is a luminal type A, a luminal type B, HER2 minus, HER2 plus. And this one is a HER2 type, HER2 positive is a receptor, HER2 receptors positive. And other is the basal-like ER, PR, and HER2 negative. No receptors over there. And based on this, based on the receptor status, the prognosis is also classified. They're classified into three groups. One is considered as the first one we can consider as a better prognosis. Other is considered intermediate types and worse prognosis. So if you look at that, the luminal A or B, this is the estrogen receptor positives are better prognosis as compared to triple negative, which is a worse prognosis. So when you find the patient with the ER positive, the chance of the disease to cure, I will say cure the disease, is very high as compared to triple negative, is worse conditions. Okay. So now, but interesting part over here, breast cancer types, although the breast cancer is the 3 different types, HER2 positive or estrogen receptor positive, HER2 positive or triple negative, but it is the best, they are very heterogeneity. You can see sometimes you can see in the triple negative breast cancer, you can see the estrogen receptor positive cells or HER2 positive cells. Sometimes you can see ER, PR, HER2 positive or triple negative with some cells are HER2 positive. So it's kind of mixed population you can see in one patient. Not always, but sometimes you can see. That's why the biggest challenge in the treatment. So, and therefore we consider that the ER positive cells could be the progenitor cells for luminal type or basal type. Progenitor cells, luminal type, this luminal type, that means the ER, PR positive cancer can move towards HER2 positives or TNBC, triple negative breast cancer. So we can thus consider this one as a progenitor cells. And based on these histological characteristics, we consider breast cancer is a mixed bag of cells. Is ER positive, it could be the HER2 positive, it could be triple negative, or it could be both, all 3 complex. So breast cancer is a mixed bag of cells and a complex ecosystem of cell populations. So that's why sometimes treatment is really difficult for us. So progenitors of the luminal A and B and HER2B cells may serve as a precursor for the TNBC cells. So now we can consider treatment option. As you know, the luminal type is basically hormone therapy, estrogen therapy. HER2 is a monoclonal antibody Herceptin therapy. TNBC is always in either HER2, Herceptin, or estrogen receptor or tamoxifen with chemotherapy treatment or chemotherapy alone. That's the treatment options. But in reality, chemotherapy does not work very well. Most of the time, they relapse, there is still loss of sensitivity of the chemosynthesis. That's the biggest problem in chemotherapies. And we always find a patient with resistant to chemotherapy eventually. So tumor initiating cells, so who plays this one? The tumor initiating cell are cancer stem cells that cannot be destroyed by none of the above therapies. Neither hormone, nor monoclonal, nor chemotherapy can destroy the stem cells. These treatments can blocks or delays the growth of the cancer cell for long period of time, 5 years, 10 years, or maybe 15 years, but it cannot completely destroy the cancer cells. So that's why you cannot make a judgment that, okay, how long, when, and these cells will relapse, the disease will relapse. It is a big challenge. Still, it is a big challenge. So that was our main goal, to find how to overcome this disease. How can it destroy the stem cells so that disease cannot come back again. Okay, now if you look at this cartoon, this is the tumor cells, and this is the CSC, cancer stem cells. That cancer stem cells basically metastasize to the distant organ like lungs, liver, or some other organ, some places, bones. Therapeutic agent basically destroys all cancers, this epithelial type tumor cells, but they cannot touch them. So, they're still there. That's why the problem is that. And again, if you come the novel agent, so we're looking for novel agents that can convert the epithelial cells, ER negative cells, to ER positive cells, or CSC, or that is a cancer stem cell, which is, by the way, this which is the estrogen receptor negative, they can convert to estrogen receptor. So we need some kind of agent that can convert the CSC to tumor epithelial cells. Sensitive to chemotherapy or hormone therapy. To make it, we need to do something. That was our goal, to make these CSC cells to convert into the tumor epithelial cells, which cannot basically form tumors or but it's not aggressive type. Okay. So we found in our studies the CCN5 WIS2 is that agent that can convert the cancer stem cells, ER negative cells to ER positive cells, and cancer stem cell also convert to the ER positive epithelial type cancer cells. So cancer cell, but not the aggressive type of natures. Let's go why. I will discuss how. So now you can ask me the question, what is a CCN5? Everybody asks this question, and now I'll show you that CCN5, which is also known as a WIS2, Wnt1 induced signaling protein 2, is a multimodular, multicellular protein and a member of CCN. Now changed the name, the CCN number, but initially it was a SAR61 CTGF knob family growth factor. They have a 4 special domain 1, domain 2, domain 3, domain 4 in CCN family. But interestingly, in CCN5, this CT domain is missing. They have only 3 domains. All other family members, they have 4 domains, but except CCN5 has only 1 domain. And they have in the promoter of the CCN family, you can say multiple site for in the promoter, like AP1, NF-kappa B, EST1, SP1, SMAD, HIF1 alpha, and etc. So it's a long story I've short briefly. Initially, 15 years ago, when he did the differential display experiments, it was now obsolete, the technique, and we used 1 and 2 human mammary epithelial cells and 3 and 4 MCF7 cell. We initially started this one when we found that this particularly WIS2 or WIS2 genes or CCN5 genes overexpressing breast cancer cells, but not in the normal cells. So we try to see whether this can overexpress in the cell line also. So we tested in the cell line. We found, interestingly, it's a very initial studies. We found that MCF7 is overexpressed, ZR75 these all our cells are overexpressed, but not the MDM231 or HE70BC. But interesting part, these cell lines, MCF7, HC70, these are ER positive cells, but HC70, MDM231, BT20, they are ER negative, triple negative breast cancer cells. So confusion developed at that time, why the cells are only expressed in the ER positive cells, not in the triple negative breast cancer cells. So I am not going to details about that, this part also. I already mentioned there's a 25-3 protein. Okay, so we tested in distribution of CCN5 WIS2 mRNA and protein in the different breast tumor samples, real life specimens, by in situ hybridization and immunohistochemistry. We found that the WIS2 CCN5 only expressed in the atypical ductal hyperplasia, the pre-neoplastic conditions, and DCIs, ductal carcinoma in situ, but well differentiated, moderately differentiated, and poorly differentiated expression of CCN5 was undetected. We didn't find that expression. So our initial thought was that CCN5 could be a positive, cancer positive genes, but now these studies gave us a different impression about the CCN5 protein. So, and then we checked that CCN5 overexpressed, through the ATCG and TCG, we found that this is called breast cancer invasive TCGA. We find that the gene expression group, the low is the green one and high is the high risk and low risk factor. In the high risk factor, the CCN5 expression was almost 0. Minus, you can look at the gene expression level is 0, while low risk breast cancer patient, they have some expression level from high to low. And also Kaplan-Meier plot find that the CCN5 positives are low risk patient, long survival rate as compared to the high risk or low no expression patient. Patient with have no expression, as overall survival rate is very low as compared to the overexpression. So that studies, that results tempted us to understand what is the function of the CCN5. We tested this one in colony formation assay. We found that in cancer cells, when treated with the CCN5 recombinant protein, that we developed this one in our laboratory, and we found that it blocks the colony formation assay. I'm not going to details. If you have any question, you feel free to ask me. I can discuss all these things. And also it induces apoptosis. In some in asynchronous population, in the some population, you find it. Next, what we did is treatment to prevent the in vitro migration. You know, the migration or sphere formation is one of the hallmarks of the invasive feature of the disease. And we found that the MDMB231 or HC70 cells, when treated with the HR mean human recombinant CCN5 protein for 7 days, and we found their the in vitro migration significantly decreased in CCN5 treated cells. Next experiment we did it, that is the different CCN5 highly expressed in both human and meat. Okay. In this regard, I want to say something about this. If you want to know, the mean model or mind model is the model we discovered. We published a recent paper last year. This mind model is we injected the particular one cell that's called the DCIS.com cells. We injected MCS DCIS.com cells into the mammary fat pad through the nipple, and very interestingly, you can see very sequential development of the disease from ductal carcinoma, atypical hyperplasia to DCIS and invasive. It will within 6 weeks. If you inject it to the mice through the nipple, this was called mice interductal, you can see the tumor growth from normal duct, and after 4 weeks, you can see the DCIS, and after 6 weeks, you can see the invasive cancer. DCIS IC means invasive cancer transition here is. And interestingly, when we did this one, we found that the only is, if you look at this is the human and look at it is the mice. In human, you can see the normal duct, DCIS with CCN5 positives, and invasive cancer, no CCN5 expression. While here you can see in mice exactly the same thing, but within a very short, brief period of times, like 4 weeks after, you can see the DCIS with the full positive cells, but eventually disappear. I mean, you can see the transition. Once the CCN5 disappear, the cells become invades to the adjacent tissues. And this has been proved by this experiment. This is called TIFF experiment, tissue fluid. And what we did here, we fresh tissue, we collected and incubated for 2 hours, and they determined the CCN5 concentration using Western blot, and we found that the DCIS has a huge expression as compared to the IC in the same tissue. That's the interesting part, and we found in the human as well as in the mind model. Next experiment, so we can see this one. Next, what we did to confirm the experiment, DCIS to IC, ductal carcinoma in situ to invasive cancer transition is promoted by a blockade of CCN5 in mind xenograft model. What we did, this is the CCN5 immunohistochemistry, both. DCIS cell with teton, we transfected the in teton system that you can, if you use the tetracycline, you turn on and off the CCN5 gene. So we injected this cells, transfected cells into the mice through mice fat pad through nipple here and here, and one group I gave dogs, one is without dogs, and we found the dogs treated mice, they stay in. You can see here the dogs treated mice, 5 out of a DCIS. It is only for 4 weeks. After 4 weeks, the 4 weeks after intraductal injection into the female node mice, we found DCIS-like structure in dogs, untreated mice. While we give it the dogs, that means we turn on the genes, turn on the HUS2. Okay, so we find here that docs, 5. So this is a shRNA. Basically, we kept. Sorry, I forgot to tell that. This is the shRNA. Why we inject the shRNA? Because to turn off the histogene here. Sorry. Oh my God.
Dr. Banerjee, we are over time, so if you could wrap up, that would be great.
Pardon? Back up.
We're over the time, so if you could wrap
so
up.
let me finish this. Okay. So here is the. Okay. So this is my whole mind model. Now I don't want to go there very details. Basically, I have to say that for all this experiment, we found that epithelial mesenchymal transactions and stemness, so local mid-invasive breast cancer, and then we give the CCL5, it blocks the whole systems, the immunosuppression also. And this is the mechanism we follow, is that is block all these experiment genes expressions. So also through the P27 expression profiling, and then also cell cycle regulation, and then also we tested in the human, in the mouse model, it blocks the tumor growth, MDM2 is the immunohistochemistry. So basically, that's the last model I will show you, that it binds with the estrogen receptor, then they go turn on the CCN5. This CCN5 goes to bind with the beta 1, alpha and beta integrin, PI3K-AKT, and then FOX, then FOXA3, then goes there, and then they block the and activate the estrogen receptor in the cells. Sorry, I mean, I could not show you all details, and timing is a factor. And also, anyway, so thank you. And if you have any question or anything, we can discuss later, if you want.
Thank you, Dr. Banerjee. I think we have time for one question, and then we'll go to the break. And please write the questions in the chat box.
Sure.
All right, I'll ask one question. Are there any chemical inducers of CCN5 that you have? Are there any chemical inducers of CCN5?
Okay, good. Because that's the I had a plan to show you anyway. The whole thing is, yes, right now we have we patented, not a drug, we pegylated the protein, the CCN5 recombinant protein. If you inject into the mice, you can reduce the tumor growth, intratumoral. But if you inject into the systemic, you cannot do that. But what we did, we pegylated the whole CCN5 protein, C-terminal areas, and now we can use this one. We recently we funded investigation fund for this. If you inject into the mice, the pegylated protein can work. In addition to that, very old things, the EGCG, there's a component of the T, can activate this protein. But the problem is EGCG is very low half-life. The half-life is very poor. That's why what we're doing, we are now doing a targeted nanoparticles to turn on the CCN5 in the breast cancer.
Great, thank you so much. We'll now turn to the break. It'll be 10 minutes, and then we'll reconvene at 11:25.
How can I give it to you now? Abby?
I think one of the hosts will redirect.
I didn't find to turn off.
If you go to the participants where you click that.
Participant, yes.
The bottom. Yes. And then if you find Maria's name, and I believe you click more.
Yeah, now I got it. Whose name is? Abby?
I think Maria already claimed host. So you
got it. Okay, never mind.
You got it?
Yeah, we're good.
Okay. Thank you.
So Abby, what time are we coming back?
11:25.
Okay, or 2:25 if you're on the East Coast.
Oh, sorry.
So thank you, everybody. Feel free to take this time to use the restroom or to stretch or to rest your eyes. And at the same time, we have a special treat for you all, and especially those who know Dr. Sarkar. I'm Sophie Sarkar, his youngest daughter. And I'm personally not a scientist, but I am an artist and I love photography. And I went into the archives for you all to get to see some really special photos of special moments in my dad's life, starting from the 1970s. So I'll be sharing this slideshow during the break, and we'll put some music along with it so you can enjoy yourselves, and then we'll come back at 25 past the hour. So you'll know when it's time to come back when the music goes silent. All right.
ললিতা গো ওকে আজ চলে যেতে বল না, ওকে আজ চলে যেতে বল না। ও ঘাটে জল আনিতে যাব না, যাব না, ও ঘাটে জল আনিতে যাব না, যাব না। ও সখী অন্য ঘাটে চল না ললিতা, ওকে আজ চলে যেতে বল না। দিবালোকে সে আমায় নাম ধরে ডাকে, আমাকে সবাই দোষে সে সাধু থাকে। দিবালোকে সে আমায় নাম ধরে ডাকে, আমাকে সবাই দোষে সে সাধু থাকে। অসময়ে সময় কিছু কেন সে বোঝে না, আমি কি তার হাতের খেলনা ও ললিতা। ওকে আজ চলে যেতে বল না ললিতা, ওকে আজ চলে যেতে বল না। নিশিরাতে বাঁশি তার শীত কাটি হয়ে, চুপি চুপি ঘরে এসে বাজে রয়ে রয়ে। নিশিরাতে বাঁশি তার শীত কাটি হয়ে, চুপি চুপি ঘরে এসে বাজে রয়ে রয়ে। যখনই ডাকবে সে তখনই যেতে হবে, আমি কি এমন কর ফেলনা ও ললিতা। ওকে আজ চলে যেতে বল না, ও ঘাটে জল আনিতে যাব না, যাব না, ও ঘাটে জল আনিতে যাব না, যাব না। ও সখী অন্য ঘাটে চল না ললিতা, ওকে আজ চলে যেতে বল না।
I wanna give. I've been a miner for a heart of gold. It's this expression I never give. It keeps me searching for a heart of gold. And I'm getting old. It keeps me searching for a heart of gold. And I'm getting old. Oh, help me somebody. I've been to Hollywood. I've been to Redwood. I crossed the ocean for a heart of gold. I've been in my mind, it's such a fine line. It keeps me searching for heart of gold. And I'm getting old. It keeps me searching for heart of gold. And I'm getting old. Searching for a heart of gold. Keep me searching and I'm growing old. Keep me searching for a heart of gold. Keep me searching and I'm growing old. Keep me searching for a heart of gold. And I'm getting old. Child, and don't cry. Your folks might understand you by and by. Just move on up toward your destination. Though you may find from time to time complication. And take the trip. Though there may be wet road ahead and you cannot slip, just move on up. For peace you'll find into the steeple of beautiful people where there's only one kind. So hush now, child, and don't cry. Your folks might understand you by and by. Move on up and keep on wishing. Remember your dream is your only scheme, so keep on pushing. Take nothing less than the supreme best. Do not obey, no mistakes forsake, but you can pass the test. Just move on up to a greater day. With just a little faith, if you put your mind to it, you can surely do it.
All right. Thank you, Sophie, for that wonderful presentation. We will continue to the next talk. Next up, we have Dr. Amkaram Gangasetti. He is a scientist at Rutgers University whose research focuses on epigenetics, and he will be talking about fetal alcohol programming of stress and immune axis functions.
Dr. Amkaran, I think you're muted.
Can you able to hear now?
Yes.
Yes. Okay. This is really a very exciting event to being a speaker at this important event, which is organized by Maria and Abhi. I need to really thank them. It's really a wonderful break. You people have shown a small video showing Dr. Sarkar's achievements, and I have been attending his award-winning ceremonies. This is my third time, but this time I'm being a speaker. It is really a great pleasure to give you a talk on this occasion. Today, I will be talking some of our previous findings and current findings of fetal alcohol programming of the stress and immune axis functions, which is done in our lab. I've been associated with Dr. Sarkar for almost 8 years. He is very passionate about the research, and also he is very aggressive in his research all the time, work and make all of us very productive. Oh, I couldn't able to move on to my slides. Oh. Yeah. As we all know that alcohol used during pregnancy is quite prevalent. The CDC reports that more than 52.6% of the pregnant women, they drink alcohol. In US, 1 in 10 to 12 pregnant women, they drink alcohol. 1 in 30 pregnant women, they admit drinking alcohol, which is the binge alcohol drinking. The fetal alcohol spectrum disorder is a condition occurring in children who were born to mothers who drink alcohol when they are pregnant. They have a deficiency of the growth, the facial abnormalities, there is a damage in central nervous system, as you see, shrinkage in the brain, there is a neurobehavioral deficits. Sometimes you get combination of these features that result in fetal alcohol spectrum disorder. In our lab, we mainly use a rat animal model for fetal alcohol exposure, for which the Fisher Fisher 344 rats, when they are pregnant during gestation day 7 to day 21, we feed them with 3 different diets. One is the liquid diet containing 6.7% alcohol, call them as AF. It is only the liquid diet, no alcohol, call them as pair fed, or otherwise the regular rat chow, they ad libitum, the AD. The offsprings, once they are born from 3 different mothers, we label these names AD, PF, and AF. The AD and PF, they serve as a controls for AF. When these animals, when they attain the age of 2 to 3 months, we use them for our experiments, mostly either by collecting the hypothalamus and sperm. In this animal model, it maintains the blood alcohol concentration during the time when we feed them with alcohol. It is 120 to 150 mg per deciliter. It represents the binge alcohol drinking. Our lab mainly focuses on stress and the HPA axis, that is hypothalamic pituitary adrenal axis. In response to any of the given stress, the hypothalamus releases the CRH, the corticotrophin releasing hormone. It acts on pituitary gland, then thereby releases the ACTH, adrenocorticotrophin releasing hormone, in secretion. Then ACTH, it binds to its specific organ, adrenal gland, and thereby synthesize and release the corticosteroid. This corticosteroid in turn negatively regulate the CRH expression in hypothalamus, thereby forming a classical negative feedback loop. Our lab has previously reported that fetal alcohol exposure can alter the stress, anxiety, and immune function. We recently confirmed these results in Fisher rats. The stress axis was measured with plasma corticosteroid levels, which is done at 2 different time points, one at basal level and 1 hour after restraint stress. We found that the plasma corticosteroid levels were significantly increased in AF animals compared to the AD and PF animals, both in case of the male and female. The anxiety behavior was evaluated by elevated plus maze test. We measured the time spent in closed arm and also open arm. The AF animals, they spent more time in the closed arms and less time in the open arm, showing that these animals have more anxiety compared to the control animals, both in case of the males and also in the females. The interferon gamma, which is a cytokine, play a very vital role both in case of the innate and adaptive immunity. Some of the immune cells, the B cells, T cells, and natural killer cells, they secrete the interferon gamma. This interferon gamma, which is secreted from the NK cells, they play a very major role in host defense mechanism, mainly to avoid or to kill any of the infections. This interferon gamma expression, we measured it in the spleen, both at mRNA level and protein level. Here I show the protein expression. This expression is significantly reduced in case of the AF animals compared to the controls, both in male and females. This results confirms our previous findings that fetal alcohol exposure increases the stress, increases the anxiety, and it reduces the immune function. The POMC, which is a pro-opiomelanocortin gene, it is highly expressed in hypothalamus. It plays a vital role in regulation of the stress, immune axis, and brain award system. This becomes a kind of greater attention for our studies. We measured the expression of the POMC in these animals, hypothalamus, F1 animals. The POMC expression is significantly reduced in AF animals compared to the controls, both in case of the male and female animals. Then the question came, how the fetal alcohol exposure reduces the POMC expression? What is the probable mechanism for this? We thought that the epigenetic mechanisms may play a role in regulation of gene expression without altering any sequence of the DNA. there are three different epigenetic mechanisms. One is the DNA methylation, histone code modifications, and non-coding RNAs. Of all the three modifications, the DNA methylation is the very prominent epigenetic mechanism which play a role in repression of the gene. Then to explain this DNA methylation in the promoter of the gene, you will have the CPG, that means cytosine followed by guanine. If these CPGs are rich, thereby forms the CPG island, then if the cytosine residues are getting methylated, then it can tend to transcriptional repression because the transcription factor cannot able to bind there. It can only recruit the certain methyl CPG binding proteins onto it. That is how this DNA methylation works as an epigenetic mechanism to regulate the gene expression. Then we wanted to see whether the POMC promoter has any CPG island. The methprime web tool we used to predict the CPG island in the POMC promoter, we found that there is a huge CPG island in the POMC promoter. We used two different methods. One is the MSP and another one is the pyrosequencing to measure the POMC promoter methylation. The MSP is the methylation specific PCR, which is a semi-quantitative method to determine the POMC methylation. We found that the POMC promoter methylation is significantly increased in AF hypothalamus compared to the controls. The pyrosequencing is a highly sensitive method which can able to detect the methylation at individual CPGs. We targeted three different CPGs and found that all the three CPGs showed increased methylation in AF animals compared to the controls in hypothalamus. Then there is a tendency that whenever there is a hypermethylated promoter, the methyl CPG binding proteins can be recruited onto the hypermethylated promoter, thereby repress the transcription. We measured the expression of the MECP2, that is the methyl CPG binding protein, both at mRNA level and protein, and we found that the MECP2 expression is significantly increased in AF compared to the controls. Then our question is whether the MECP2 is enriched at the POMC promoter, then what would be its level of enrichment with the alcohol exposure? We performed the chip assay to determine the MECP2 enrichment on POMC promoter. We found that MECP2 enrichment is significantly increased in AF hypothalamus compared to the controls. All this data clearly suggests that the fetal alcohol exposure, it programs the hypothalamus to reduce the expression of the POMC by hypermethylating its promoter and recruitment of the methyl CPG binding onto its promoter. The next question came in whether this fetal alcohol exposure induced adverse effect on POMC, will it be transmitted to successive generations? Then our lab came up with very exciting publication in 2012, which got quite a great attention in the field of alcohol research, and it got more than 100 citations. The answer is yes, the fetal alcohol induced adverse effects on POMC is transmitted to successive generations through male germline. However, this study was conducted in outbred Sprague-Dawley rats. It raises the question that any of the genetic variants may play a role in its transmission. To address this question, now we started studying this in Fisher rats, which are the inbred rats, for which we used a very unique strategy to develop both the male germline and female germlines of the F2 and F3 animals. As I already mentioned how we can generate the F1 animals both from alcohol-fed mothers and pair-fed mothers. And here these are the animals which we consider it as F0. These are the only animals which drink alcohol. Then the rest of them, they were only exposed to the alcohol, whereas in case of the F1 animals, they were exposed to the alcohol during their fetal development. Then to develop this male germline, what we take it is, we take this male alcohol fed animals, that is the AF males were bred with control females to generate the F2 animals. Then in F2 animals, we get both male and female offsprings. then the male offsprings were used to bred with control females to generate the F3 animals. That means both in case of the F2 and F3, you get this male germline, which would be denoted as AFM, that is the male after AF, that is male germline. It will have both male and female offsprings. Whereas the female germlines were generated by taking the AF females bred with control males to generate F2 animals. Then in F2 animals, the AF females were bred with control males to generate F3 animals. Here also you get both male and female offsprings, but they are female germline. You represent them by adding F after AF. That means whenever you say AFF, it is the alcohol-fed female germline. AFM is the alcohol-fed male germline. You will have both the offsprings. In the similar fashion, we generated the pair-fed animals also, both male germline and female germline. For the AD, it doesn't come any difference. All of them were fed with the rat chow. Then our next question, we started measuring the stress axis and also the anxiety behavior. The plasma corticosteroid levels, after they were induced with restraint stress for 1 hour, we found that the levels were significantly increased in male germline. There is some level of increase even in case of the female germline, but it is more prominent in male germline in case of the F2 animals. Whereas in case of the F3 animals, we found that the plasma corticosteroid levels were significantly increased only in male germline, but not in female germline. The anxiety behavior, which we measured with the same elevated plus maze, we found that the anxiety behavior is significantly increased in F2 male animals, only in male germline, but not in female germline, by showing an increased time spent in closed arms and less time in the open arm. Whereas in case of the F3 animals also, whereas in case of the, this is the F2 animals, sorry. Whereas in case of the F3 animals also, we found the similar trend that the anxiety behavior is significantly increased only in male germline, but not in female germline. This data clearly suggests that we could able to confirm this transgenerational inheritance of fetal alcohol exposure induced stress axis and anxiety behavior is transmitted through successive generations only through male germline. There is some change at the plasma corticosteroid levels in F2, but not in F3. Then, as we know that the POMC, which is the master regulator of the stress axis, then we started measuring the POMC expression. We found that the POMC expression is significantly reduced only in case of the male germline in both F2 animals and also in case of the F3 animals. Then the epigenetic mechanism, which we measure by promoter DNA methylation, which the pyrosequencing data clearly shows that out of 3 CPGs, there are 2 of the CPGs showed its increased methylation only in male germline, but not in female germline in both F2 and F3 male hypothalamus. This clearly suggests that the fetal alcohol-induced adverse effect on POMC can able to transmit to F2 and F3 generations only through male germline. Even in the Fisher rats, which are the inbred animals, this rule out the possibility of any genetic variants playing role in this transmission. It came a big question like, what is it really playing a role for this male germline transmission of these POMC adverse effects? Then since the transmission is only observed through male germline, we were interested to see whether there is any change in the expression of the POMC in both F1, F2, F3 animals. We found that the POMC expression is significantly reduced with alcohol exposure in case of the sperm samples in F1 animals, whereas in case of the F2 and F3, we only observed POMC expression is reduced in male germline, but not in female germline. This data pretty much coincides with similar data which we observed in case of the hypothalamus, suggesting that there might be something in the male germline playing a role in this transmission, fetal alcohol exposure induced transmission on POMC. Then it came to our mind, then the Y chromosome is the one which is distinct in case of the males and females. As we all know that in case of the females, you get 2 X chromosomes. Only in males, you get 1 X and 1 Y chromosome. If you look at the structure of the Y chromosome, then you have the distal region. At the distal region, you have the paired region, which pairs with the X chromosome. Then rest of the region is the non-pairing region that mainly transmit from fathers to the son. The SRY is one of the non-functional gene which is involved in male reproduction, and it is mapped to this N-pair region, that is the non-pairing region of the Y chromosome. There is a study which came in 2011 reported that the beta endorphin, one of the functional peptide of the POMC, it is regulated by the N-pair region of the Y chromosome that comprises the SRY. There is also another study came in the next year, that is 2012, SRY regulates the endocrine response to the stress. I think this prompted us to think that there might be some role of SRY in regulation of the POMC, and we prompted to investigate whether SRY may play any role in regulation of the POMC. Then first thing, we measured the SRY expression, both in case of the hypothalamus and sperm. We found that the SRY expression is significantly reduced in hypothalamus in F1 animals. Then the SRY expression is only reduced in male germline, both in case of the F2 and F3 animals in hypothalamus. The similar trend was observed even in case of the sperm. This results shows pretty much same trend as we observed for POMC, both in case of the hypothalamus and sperm. It suggests us there might be some kind of interaction between this SRY and POMC, which we wanted to investigate further. Then what we did was that we knocked down the SRY both in vitro and in vivo. Using the antisense oligos for SRY, we transfected them in POMC neurons. These oligos effectively reduced the SRY expression. To our surprise, we found that this POMC expression is also significantly reduced when you knock down the SRY. Then we wanted to see whether we get the same effect in vivo. We did the ICV infusion of these oligos in rat brain. Then we collected the hypothalamus and measured the SRY expression. The SRY expression is significantly reduced with the antisense oligos. At the same time, we noted that the POMC expression is also significantly reduced by knocking down this SRY. What these results clearly suggest that SRY may have some role in regulation of the POMC. Our next question is it the SRY acts as a transcription factor, binding it to the POMC promoter and thereby regulate the POMC? To address that question, first, we used some web tool to predict is there any SRY binding sites at the POMC promoter. We used one of the web tools, that is the transfact, predicted certain SRY binding sites at the POMC promoter. Then we started looking to see whether the SRY is enriched at the POMC promoter using the primers flanking these SRY binding sites. We found that the SRY is pretty much enriched at the POMC promoter. This SRY enrichment is significantly reduced with alcohol exposure in hypothalamus. This clearly suggests that SRY may be recruited onto the POMC promoter and then regulate the POMC gene expression. We just next wanted to ask the question whether the SRY, is it really binding it onto its binding sites at the POMC promoter and thereby promote the POMC expression? Then, as I mentioned, that the web tool which we used to predict the SRY binding sites, there are 3 different SRY binding sites were observed in POMC promoter. We cloned the POMC promoter region which contain all 3 SRY binding sites into a reporter vector which is upstream of the firefly luciferase gene. Then we measured the luciferase activity as a measure of the POMC promoter activity. What we did it is, We took all these plasmids. It is just the basic reporter vector, the vector containing the POMC promoter. And here it is the vector containing POMC promoter was transfected along with empty vector or the vector which will have the SRY open reading frame. That means the minute we overexpress the SRY, it actually increases the luciferase activity, which is a measure of the POMC promoter activity, significantly compared with the vector control. This clearly suggests that the SRY may be binding it to its SRY binding sites on the POMC promoter, thereby promoting this SRY expression. Then our next question we were asking is that whether these binding sites are really essential for SRY to bind it onto the POMC promoter, thereby promote this POMC promoter or POMC activity. Then what I did was that we mutated these SRY binding sites in two different combinations. One is a single mutation. The mutant 1 and mutant 2 are the single mutation. Then the mutant 3 was generated as a double mutation. All this was done using the site-directed mutagenesis. Then we performed the same assay luciferase activity as a measure of the POMC promoter activity. Here we introduced these plasmids, either the wild type plasmid containing the POMC promoter, which is the wild type, or the POMC promoter, which is the mutant in which one binding site is mutated. The mutant 2 is also a single mutation, and mutant 3 is a double mutation. Then along with these plasmids, we introduced either empty vector or the SRY overexpression plasmid. As usual, in case of the wild type plasmid, which has the wild type POMC promoter, the SRY promotes the POMC promoter activity, as we know. Then we found that even in case of the mutant 1 and mutant 2 also, the SRY is able to promote the POMC promoter activity. This clearly suggests that it might be utilizing the other two transcription factors, the other two binding sites of the SRY to promote the SRY promoter activity. I think the mutant.
We are running a little bit late.
Yes,
so
yes, I'm done almost. Yes. In case of the mutant 3, the double mutant, when we mutated two of the binding sites, the SRY cannot able to promote the POMC promoter activity, clearly suggests that the SRY requires at least two binding sites to promote POMC promoter activity. To summarize our data, the fetal alcohol exposure, it actually increases the stress axis, increases the anxiety behavior, reduces the POMC expression, increases the POMC methylation. All these adverse effects induced by fetal alcohol exposure, it is transmitted to F2 and F3 generations through male germline via the sperm. The SRY, which is mapped on one Y N pair region of the Y chromosome, under normal conditions, it can bind to the POMC promoter through its SRY binding sites and activate the POMC gene expression. Once with the fetal alcohol exposure, there is a promoter hypermethylation. Then the SRY recruitment is significantly reduced. Then the methyl CpG binding proteins are attracted towards the hypermethylated promoters, thereby reduce the POMC gene expression. Our next future aims is this SRY regulation of POMC transmission. Will it be continued for successive generations is our future plan of research. I wanted to thank Dr. Deepak Sarkar for providing this opportunity to work in his lab for almost, my journey is with him for almost 8 years. He always enjoy working with students. You can see his smiling face, and he always direct us to go how we need to be highly productive and make success. Here I wanted to acknowledge some of my previous and current colleagues who contributed for this project. Here is the Andy Med, who contributed for SRY, and Ajay Palgani did cortices, and the Shaista Chowdhury did anxiety behavior. Here are our past members and the present members of our lab. And I thank Dr. Deepak Sarkar for all the support through NIH grant funding.
Thank you, Dr. Gangaseti. That was very interesting talk. We're running a little behind time, and there are 3 great questions in the chat room. If you could address those when you have time, we'll move on to the next talk.
Okay. Can I stop the share? Abhi, can I stop the share?
Maria, could you stop this? Okay. All right. Our next speaker is Dr. Jonder Rangunathan. He is the Program Director for Division of Neuroscience and Behavior and at the National Institute of Alcohol Abuse and Alcoholism at the National Institute of Health. He has held this position for 10 years, and he will give us more details about that.
Hello everyone. Can you hear me?
Yes, we can hear you very well.
Okay. Thank you very much. And first of all, let me thank Maria and Abby for inviting me to this very interesting event. I know Dr. Sarkar for over 10 years since I've been at NIAAA. I managed several of his grants over these years. He has been a very productive scientist, and he made very good contribution to alcohol research. One of the things I just want to highlight briefly, quickly, is that when I started at NIAAA almost 10 years ago, I was assigned one of his grants, which is a merit award. This is a very prestigious award given to established scientists who has done very innovative work on alcohol research. This means you get an award grant for 10 years and you don't need to submit any application. It is continuous. And he got this award from, I think, up until 2019. It just ended and he's trying to resubmit as a new R01. And I'm sure he will get it also. So that was a big honor for him. And I was happy to be associated with his merit award and several of his other grant applications. So I'm sure he well deserves this honor from New Jersey Academy of Science. With that, I will move on to my presentation. So today I'm going to talk about some of the priorities at the NIAAA with the Division of Neuroscience and Behavior. And I just briefly highlight some important initiatives we are working on. And also then in the second part, I'll talk about NIH grant funding in terms of the process of various grant mechanisms. Of course, this is a very big, vast area, and I try to cover everything within like 10 to 15 minutes. So to start with, as we all know, alcohol is one of the ancient toxic agents, and it has been used for thousands of years as the known, like right from about 5,000 to 8,000 BC. And very early on, like about 400 BC, they identified some of the negative or harmful use of alcohol. But later on, only in 11th century, there was really a clear explanation how alcohol can cause liver inflammation. So this was the whole history, but now we know in the US, I don't know how to move this. Oh, okay. Alcohol has mixed health effects. So at low doses of alcohol can have a beneficial effect. However, long-term high use of alcohol has severe effects on the whole body and also in the brain. Indeed, alcohol is a leading risk factor for various coexisting comorbid conditions. In the US, about 30 to 40 million people drink too much of alcohol. This is the recommendation from CDC and also from NIAAA about the excess drinking limits. I mean, you can read this, and this is all in the website. But for now, this also drinking too much includes for some population is actually no drinking. For example, in pregnant women, no alcohol use is recommended. And also anybody under the age of 21, the alcohol use is not recommended. So this is the current state of alcohol consumption in the US. And the alcohol, NIAAA was started almost 50 years ago. And its mission was to generate and disseminate fundamental knowledge about the effects of alcohol. So this include conducting and supporting alcohol-related research, coordinating and collaborating with other research institutes and federal program, collaborating with national and international organization, and also the translating the research finding to healthcare providers, researcher, and policymakers and public. This was the mandate for the NIAAA. So currently, NIAAA look at this as alcohol use across the lifespan, right from fetal alcohol exposure all the way to seniors and old age population. So we look at the prenatal alcohol exposure, the fetal alcohol exposure, and then the alcohol exposure to children and adolescents, adolescent binge drinking, which moves into young adult and middle age. Some of them become alcohol dependent. And then at the old age, alcohol use, even at the moderate amount, could interact with the medication they are taking. In fact, now we have a lot more data showing that how early alcohol use can be also a risk factor for Alzheimer's disease. So the use of alcohol across the lifespan has significant effects at each age group. So this lifespan theme transcends all aspects of science, the neurobiology, epidemiology, epigenetics, genetics, and metabolic aspects. Now, we have this strategic plan for every 5 years, and the last 5 years ending next 2021. This is some of the priorities where NIAAA is working on. And you can look at this, the details by the strategic plan. It's available on the website. You can download and read all the details about it. But I just give the short summary of different aspects of the strategic plan. I don't want to go read it, but now I focus more on the neuroscience because I work at the Division of Neuroscience and Behavior. So this is some of the areas which we are focusing on in the brain. So identify the neurocircuits and neuropharmacology and neurochemistry that underlies the alcohol physiological and behavior effects. So explore the alcohol effects with the CNS at many levels. For example, molecular, cellular, and circuitry level, to structural changes, and to cognition and behavior. So the cognition include learning, memory, tolerance, and other cognitive behaviors. So this is the current conceptual framework in which alcohol affects the brain. This is in a review by George Coop, our director. So this is the 3 stages of alcohol addiction as he describes. So starting from binge intoxication early on, and then there is a withdrawal period which causes the negative affect and causes stress and anxiety and reward stimulation. And then there is a phase of preoccupation and anticipation which feed back into the binge drinking. And each stage, this either through stress and reward system or direct effect of alcohol on the brain, it could affect the neurocircuit or molecules or synaptic changes, causing neuroadaptation. So currently, we are using this model as the basis to study various effects of alcohol in brain. Some of the high priority areas of NIAAA currently are alcohol, stress, and PTSD and traumatic brain injury, because this is a very important area. And then alcohol and neuroimmune interactions, pain and opioid and alcohol use disorder. This came to the forefront now because of all this opioid crisis. And people with using opiates for pain, they also use alcohol during that time. So this kind of interaction between opiate and alcohol during pain conditions. And then the other important area is the alcohol and sleep regulation. We all know that alcohol can put you to sleep acutely. However, if you drink alcohol too much over time, it causes sleep dysregulation. So to understand this interaction between sleep dysregulation and alcohol use. And then finally, the alcohol and the aging brain. Again, this is also an important area now because of the recent evidence showing that chronic alcohol use causes various neurodegenerative disorders, including is a risk factor for neurodegenerative disorders, including Alzheimer's disease. And then also the functional genomics. This is something we also interested in, looking at the genetic variant related to candidate gene expression at the cellular molecular behavior level. This also including the epigenetic changes. Now, our division also has a lot of these initiatives are in the form of consortiums. These are cooperative agreements working towards a common theme. So this is a consortium of several institutions participating in a consortium format. So we have one on the genetic consortium, and there's one on integrative neuroscience initiative. We have two consortium on that, looking at one on the neuroadaptation, neuroimmune aspects, other one looking at the genetic variation, contribution of stress in promotion of excessive drinking. And then there's one on the neurobiology of adolescent drinking in adulthood called NJAS. This is to look at the effect of adolescent alcohol exposure and how it affects the adult brain in terms of the structure and function. And this is in the animal models. In fact, I had to mention here that Dr. Deepak Sarkar is an external advisory board for this NJAS consortium. And also there is another consortium looking at similar thing in a human brain. It's called NJAS. So this is a multi-site longitudinal study using neuroimaging and neurophysiological measures to study the impact of child and adolescent alcohol exposure on the developing brain. This is a long-term project that's going to go on for about 15 to 20 years in longitudinal study of these kids. So these are some of the very focused consortium looking at specific aspects of alcohol use disorder. Now, the another important area we are now focusing on is the alcohol use and comorbidity, because more than 40% of the people who have alcohol use disorder also have other psychiatric diagnoses, and more than 20% use another drug along with alcohol. And among the psychiatric disorder, more than 1/4 of them use alcohol, abuse alcohol. So this comorbidity is a very important factor now. So some of the areas we are focusing on, one is the comorbidity of AUD with PTSD and other psychiatric disorders. Other one is the mechanic studies of pain and AUD. Chronic pain and alcohol use disorders is a very interactive phenomena because people with chronic pain use alcohol to alleviate pain, which causes alcohol use disorder actually. So that kind of interaction. And the other one is the alcohol and traumatic brain injury, whereas here people who has this traumatic brain injury has shown increased susceptibility to alcohol and other drugs of abuse. And alcohol and other drugs of abuse, and then genetic factor that influence all of this comorbidity. These are some of the focus of the comorbidity area. Now, the NIAAA mission also include developing novel medication to treat AUD. Now, I don't want to go into too much of the background. Now, currently at the basic neuroscience level, we are focusing on some of these targets based on this mechanism, neurobiological mechanism. So the 3 stages of addiction I explained earlier, so you can target each stage with different medication or different mechanisms. So that is currently the focus, like binge drinking or anticipation, or the this is the stress reactivity and so on. So this is the kind of a new way of thinking how to treat the AUD, looking at these different mechanistic endpoints. So these are some of the promising drugs that are currently in different stages of development, or more like in a research stage. Here, this is all based on those previous slide I showed you about different stages of addiction. I don't want to go read everything, but this gives you an idea how this is evolving now as for the treatment for AUD. So our division, we are the basic neuroscience division, so we don't directly interact with the drug development area, but we contribute to the target identification and animal model studies, because NIAAA is a division of medication development. They have different programs like SBI or the small business program or human laboratory screening programs for alcohol use, and also clinical investigation group. So basically, we initiate these targets and molecules using molecular targets and animal models, and then they take it to human laboratory studies and also, if successful, to the clinical trial. So this is how we contribute to the medication development for AUD. So with that, I move on to the next part of my talk. And this is about the grant funding initiative at resource at NIH. This is a big topic. I can spend like 3-4 hours on this, but today I'm trying to do it in 10-15 minutes. So it's going to be very condensed, but you can look at a lot of this information, the thing that's in NIH websites you can go to for all the details. So the career stages of funding normally start from graduate student. Like you have a pre-doctoral training. There are specific programs. You can be part of a T32 training program or F30, F31. And then you move on to postdoctoral training, like F30, F32. And then there is a pathways to independence, a part section where you transit from postdoc to academic transition, and that will be the pathway to independence awards. And that's called a K awards, so different K award mechanisms. And then you go into independent award like R mechanisms. Now, so the fellowships, there are several grant mechanisms, like starting from F30, F31, F32, and F99K00. I will explain that a little bit more. I'm trying to focus this primarily for early stage and young investigator. So I'll spend a little bit more time on these F and K mechanisms. And K mechanisms are the career development, that is the transition period. And then there are research project grants, which are like R mechanisms. And NIAAA, we also have other mechanisms like P mechanism, which is the centers that are focused on a big projects centered around a particular university lab. And then we also have this cooperative agreement like UO1 that like mentioned earlier on this consortium grants. So if you start as a graduate student, you can go through different pathway to go to get an R01. This is the multiple pathway leading to the independent researcher. Either you go through F31, independent F31 or a T32 fellowship, and then you go to F32 from postdoctoral fellowship. And from F32, either you can go through K99 R00 through R01, or you can do K01 and then to R01. I will explain what this each one means. So F30 is a pre-doctoral fellowship. So this is for the US domestic institution, and it's a pre-doctoral dual degree program. So you can have a MD PhD program in this. So you can get for that, money for that. And then F31 is a pre-doctoral fellowship. This is for the PhD programs. And there are also other forms of F31, like a diversity F31 comes through the diversity area. And then there's the F32 postdoctoral fellowship. And this is, all these programs are offered by all institutes, not only NIAAA, all NIH institutes. And there are also some specific programs, like for example, Brain Initiative as an F32 program, which is specific. And then there is this F99K00, which kind of translate from fellowship to a K phase through this program. Now, for the earlier career awards, so we have K99R00. This is the pathway to independence award. So it has two phases, K99, which is a mentored phase. So you have a mentor in a particular university lab, and then you get an independent phase, which is the R00 phase. Only thing is this, to be eligible for this, you have to have a less than 4 years of PhD, postgraduate degree experience. So if you are beyond 4 years, you can apply for this. But one good thing is you can apply, if you are US citizen or non-citizen can apply for this. And if you are more than 4 years after your PhD, then you apply for a K01 award. This is a mentored research scientist career development award. And this is only restriction is that here you should be less than 10 years of postdoc experience at the time of application. And this is only for US citizen permanent residents. Then there is another one which basic scientists use, which is a K08 award. This is a clinical scientist. This is a MD or PhD person can apply, but it's more oriented towards a clinical research. There are also other K mechanisms which I have not described here. There are so many other K mechanisms like K23, K24 and all. You can look it up. But I'm just giving you only a few more important ones that's relevant for most people. So this is kind of a timeline where you start like a graduate with the F30, F31 or a T32 and go to postdoc as a F32 or F99K00. And then you move on to faculty, you apply K01, K08 or K23. So in terms of the R, there are so many different R mechanisms where established investigator can apply once you are a faculty. The R01 is the most common one. It can be investigator initiated or in response to a particular PA, and this is usually for 3 to 5 years. R03 is a small grant program. It's only very limited support. It's for any pilot feasibility studies. It's only 5,000 for 50K per year. Only some institutes use this R03 mechanism. Then R21 is exploratory developmental research. This is again a early stage project development kind of award. It's also a small award. It's only 275K for 2 years. So people normally, most cases you can try R03 first if you don't have any preliminary data. And if you collect data, then you apply for R01. On the other hand, if you have a wonderful groundbreaking idea, you want to find out whether it works or not, you can do this R21, which is kind of a high risk, high reward kind of grant. So then briefly, I talk about the process, the funding. So here, this is the different stages of application processing at the NIH. So when you submit the application, the PA submit the application, first it goes to the review, study section review. This is for the scientific merit review. And then you get a score or you may not get a score. Then it go to the council for a council review. So each institute has an advisory council. They review application and recommend funding or approve funding. And then the program officer in the institute, based on this study section and council review recommendation, recommend a particular application for funding. And then the IC director of the institute, finally make the funding decision. So these are the three, so we call it 3 stages of review: scientific review, council review, and then the program review. So you notice that all of these are recommendations. None of us, the study section or the council or the program decide funding. Only the IC director of the institute make the funding decision. Now, this process can take about 9 to 10 months. And then if you got a good score, if you recommended funding, you get funding. But it's not really 9 months. If you think about preparing your application, you actually really have to start much earlier than that. So this is kind of approximate timeline recommended by NIH. So you have to start like at least 4 months before your institution deadline for fund submission. So you plan it for 2 months and writing, and then 1 month for getting feedback from your program officer or also from your colleagues. And then you check everything is in correct order, and then you submit the application. So that's about 4 months. And then it goes up to another 9 months. So if you get funding at the first round, from the beginning, it will take about a year. And if you have to resubmit the application, if you don't get funding the first time, then it takes another 9 months. So it's really a very long process. So you have to be prepared for that. Now, in terms of the preparing application, you have to talk to your program officer first. So when you are developing your idea, talk to the program officer about the program priorities, whether the idea what you are proposing is fitted with the institute's priorities, and what kind of funding mechanism will be appropriate for this. And you can ask about any new initiative specific for your idea. All of that, program officer can give you a better idea. And then request funding institute, which institute your publication is going to go. And also you can request a particular study section, review committee in your application. Now, one question is how to find your program officer, because you are just starting and you don't know whom to contact. So here, normally based on your topic of research, if you go to the particular institute website, for example, if you go to NIAAA website, it lists all the program officers and their expertise, scientific expertise. So if you find someone which has similar expertise is like what you are proposing, then you contact that program officer. If you think no one has a similar expertise, you just contact any program officer and he or she will direct you to the correct person. So that's the first thing you want to do. Now, when to talk to the program officer? Then after your application is reviewed, the summary statement will be released, then you talk to the program officer about that. What is the positive and negative aspects of the summary statement? And then what about the funding potential based on the score? And most likely you may want to resubmit, then you have to talk to the program officer about the resubmission, how to address some of the criticisms in the review. And even after the application is funded, you have your grant, you have an award, still you want to talk to the program officer later because there will be ongoing, they will be checking the ongoing progress, or if you have a great publication, you want to communicate with the program officer, or if you have any problem or issues with your project, you want to talk to the program officer first. And also, he can also advise you on any administrative supplement, and also on no cost extension after the end of the funding period. So that's the role of the program officer, like me. Now, so I mentioned this before, what to do. They start with the program officer, understand the process, and read the funding opportunity announcement. This is very important. You have to read it before you are preparing your application. And if you are resubmission, address the review criteria, how reviewers will review your application. And if you are a fellowship or you are applying for a K application, you have to have a proper mentor for your application. Some other things which you don't want to do is ignore the institute program priorities. That is, don't submit a proposal that the institute is not interested in. That means even if you get a great score, you may not get funded. And then second thing is make sure you have the proper expertise in the area where you are proposing. And do not propose something just been done for the last 50 years and doing the same thing. That's no more. You have to have a very innovative idea now to be funded. And don't assume that ideas speak for themselves. This means you have to explain your idea properly in the application because reviewers may not know every detail. And omit background information. This is very important. Nowadays with all the electronic, you cite a lot of references, and I know reviewers will go back and check it. So it has to be proper reference from that particular context. And also don't ignore reviewers' comments on weakness when you resubmit your application. So now NIH is supporting this next generation of biomedical scientists. So there are a lot of programs or incentives for them. So for example, any early stage or new investigator, we are required to fund up to 25th percentile. So they have much better chance of getting funded. They also provide additional support for mid-career investigator. This means people who had R01 already and trying to renew it, we have to give some preference for them to continue the work. So the current emphasis on NIH funding program aimed at the early stage and mid-career investigator. Also, you can look at the NIH website for the research training and career development website, and here you can get a lot of information. And NIAAA specifically encourages a lot of postdocs to apply for NRSA F32 application. We get very few F32 applications now, so we want to improve that. And you apply for R21 only if you are planning to generate data that can lead to the submission of R01. Otherwise, apply for R01. Because R21 is not, there is the impression that R21 is easy to get than R01, but that's not true. So I have a real justification for writing an R21.
Dr. Ramanathan, we're running a little late. So
I'm almost there. Okay.
Thank you.
So this is basically, there are also other opportunities besides NIAAA, trans NIH thing. You can look it up, and I don't want to go through each one of them. One of the things I want to point out that the HEAL initiative, the second point, this came out of that opioid crisis. So there are a lot of funding opportunities available for people doing opioid use disorder research and pain research to develop non-opioid pain medication to treat chronic pain. So that will be very useful. There's also other research opportunities which NIAAA participate in these trans NIH efforts. Now, if you want to where to find information, you can subscribe for these NIH newsletters, which it comes out every day, NIH Guide, and you can subscribe. That describes all the new initiative, new funding opportunity announcement that comes every day or every week. So with that, I thank everyone for listening. And if you would need any more information, you can always contact me. This is my email address at NIH. Thank you.
Thank you so much. We're going to take a short 5-minute break. If you have any questions, additional questions for any of the presenters, please contact them directly or contact them in the chat room. Thank you.
All right, so we're taking a 5-minute break right now. And so feel free to rest your eyes, stretch, whatever you need. And if you stick around, the second presentation showcases my dad's key research findings, publications, and collaborators from 1975 to the present. And it's just a nice history to get us all prepared for his award ceremony after the break. So we will see you soon.
I never met a girl who makes me feel the way that you do. You're alright. Whenever I'm asked who makes my dreams real, I say that you do. You're outside. So be fine. Oh, fun. Look out, baby, 'cause here I come. And I'm bringing you a love that's true. So get ready. So get ready. keep on riding, riding on through. All the world needs no more, baby. Come on, start a love train. Don't need no tickets. Come on, join us. Ride, ride this train, y'all. Love train, love train. All of your brothers over in Africa. Tell all the folks in Egypt and Israel too. Please don't miss this train at the station. 'Cause if you miss it, I feel sorry, sorry for you.
All right. Hello, everyone. We're back. It's next my pleasure to introduce Dr. Shuli Chang, who is the director for the Institute of Neuroimmune Pharmacology at Seton Hall University and the president of the Society of Neuroimmune Pharmacology. She has known Dr. Sarkar and been a close colleague and friend for the last 15 years, and she will introduce our award recipient.
Where's the plate?
It didn't arrive in the mail, so we're gonna have to make it up here.
Oh, okay. Okay, slideshow. Oh yeah. Okay, it's really my greatest honor being asked to present to Dr. Deepak Sarkar the New Jersey Academic Science Inaugural Advance of Science Award 2020. Dr. Sarkar, here's your award. And also it's really my wonderful joy to present to you Dr. Sarkar's multi-seminal discovery in various biomedical fields, including the impact of access alcohol use on endocrine and neuroendocrine regulation of cell function and metabolites. His wonderful, exceptional, wonderful career actually traced back in 1976. The first publication in Nature, 1976, during his study at Oxford University, helps to establish that neurovascular communication is critical for maintaining homeostasis in the endocrine system. This publication has been cited close to 550 times by others. And the next outstanding work was Dr. Sarkar's postdoc research at the Michigan State University. Dr. Sarkar demonstrated that dopaminergic neuron dysregulation with the development of prolactin secreting tumors. This critical contribution of the Dr. Sarkar during this postdoc career and other studies led to the development of dopaminergic agent as a therapeutic agent for the treatment of hyperprolactinemia of the prolactinomatosis. And this work was published in 1982 on Science, and the work was cited more than 200 times. Dr. Sarkar and his fellow then show that the TGF-beta-1 system is a critical growth inhibitor of lactotropes. During his following his postdoc career, Dr. Sarkar, like everyone else, has established his own research teams His team discovered that fetal alcohol exposure induced hyperdynamic neuronal death by apoptotic mechanisms. This discovery has extended across multi-other alcohol pathology studies, including the brain. And Dr. Sarkar's team discovered that fetal alcohol exposure caused loss of hyperdynamic beta endorphin neuron through the microglia oxidative stress and the cytokine production, lowering the feedback of inhibition of hyperdynamic pituitary adrenal HPA axis that would sensitize the hyper-stress response in fetal alcohol exposure offspring. His team has published several articles in this research area, and critical example is that one published in Endocrinology in 2007. Dr. Sarkar's laboratory discovered for the first time that inner immune system is governed by an internal clock system and that maintain a circadian rhythm. This circadian drawing, this schematical drawing shown here shows that the circadian oscillation of the clock gene, cytolytic factor, and the cytokine in the rodent natural killer cells. Dr. Sarkar's laboratory was the first who showed that fetal alcohol exposure changes the body's clock, disturbing circadian endocrine and the immune functions. The recent data indicate that the clock gene have a major role in controlling alcohol drinking behavior and in promoting cancer. Dr. Sarkar and his team discovered transgenerational fetal alcohol exposure induced epigenetic modification of the POMC gene are inherited through the male germline for at least 4 generations. This is quite striking. And this is the key discovery require reinterpretation of a genetic study on alcoholism. Dr. Sarkar's team found for the first time that offspring from mother exposure to alcohol preconception has increased level of stress hormone and in their blood in response to an immune challenge and changes in the expression of epigenetic profile of gene that play a role in regulating stress response in their brain. And this finding suggests that epigenetic change in the mother as a result of alcohol misuse before conception may be passed on to her offspring and could have a lifelong effect on a child's response to stress. And Dr. Sarkar and his colleague also discovered that fetal alcohol exposure increased breast, prostate, pituitary, and liver tumor, and connected them to the neuroimmune dysfunction. And Dr. Sarkar and his colleague also discovered alcohol increased dimerization of the mu and delta opioid receptor. His team also used combined opioid receptor agonist and antagonist treatment to reverse alcohol immune and cancer susceptibilities. Dr. Sarkar's laboratory also pioneered the use of a beta endorphin stem cell therapy in fetal alcohol exposure offspring. They developed a novel technique to stem cells and the beta endorphin neurons. Both of them transplanted into the brain and the rats. These beta endorphin cells reduce the fetal alcohol exposure stress hypertherapy responses and suppress cancer growth. In addition to all this, when I told you very quickly show you the research that he has done, and you can see that Dr. Sarkar's study has a great impact on the fetal alcohol fetal alcohol disorder syndrome, and has been highly recognized by NJAS, as Dr. Sander has mentioned. He received a Merit Award from NJAS NIH. And his grant support and the number of the peer-reviewed papers are outstanding, of course. And further, a search of the web sites that indicate Dr. Sarkar has an outstanding record of being cited by others in the field. His index, the H-index, which is an index of his productivity and the contribution to the field, is placing him among top scientific contributor. And every paper that I just highlight a few of them, has averaged over 25 citations. Of considered note is the one that his recent paper, the one indicated transgenerational epigenetic effect of fetal alcohol on the hypothalamic POMC gene, published in Biological Psychiatry, has selected one of the top 10 to 15 top ranking articles in the Biological Psychiatry. This paper was nominated for the Z-Kind Summit Research Award. Dr. Sarkar has received many awards, and just name a few here I list. That is Melon, the Scientific Research Award from the Association of Science in American, Federation of Indian Physiological Society of Erection, the silver, the Margetti Memorial Origin Gold Medal, and one, because we talk about alcohol research, I like to call your attention that the Distinguished Research Award by Research Society of Alcoholism. And of course, that Dr. Sarkar has the very distinguished title, board of a governor professor of Rutgers University. So with this, in addition to this very outstanding scientist, Dr. Sarkar is also exceptional, kind, mentor, colleague, even not me a colleague that is more congenial, conscientious, and willing to share his knowledge with graduate students and fellow researchers from around the world. Dr. Sarkar has a sustained long-term commitment to conducting alcohol research and has made the important discovery on the molecular mechanism of alcohol pathology. And as I mentioned, that the transgenerational epigenetic study really shift the paradigm of alcohol research. When we talk about fetal alcohol syndrome, that the concept has been changes our understanding. And also, as knowing Dr. Sarkar for past 10, 15 years, then I enjoy so much that see how he is engaged to get the research and dissemination in the New Jersey state. So it's really our privilege for our society that to award such a scientist this first, the New Jersey Academy of Science inaugurated Advanced of Science Award. I hope you noticed that I put the olive leaf on this slide. Congratulations, Dr. Sarkar.
Thank you, Dr. Chang, for such a heartfelt, kind speech. We're going to move on to a list of former trainees and colleagues of Dr. Sarkar, and I'll just say a few words each. So first up is Dr. Xiaodong Chen, who's the director of the Institute of Clinical Immunology at Shanghai University of Traditional Chinese Medicine. Dr. Chen, are you there?
Yeah, I'm here.
Dr. Chen from Shanghai University.
Yeah.
I think it's like 2 a.m. there, so we might have lost him.
All right. We'll move on to Tina Franklin, who's a research scientist at Georgia Institute of Technology.
Hi, everyone, and congratulations again, Dr. Sarkar, for this well-deserved award. So I was told to keep it short, so I will keep it short. And I'm putting my timer to make sure that I don't go over time. So I figured one thing I should do is share a short anecdote with you guys on how Dr. Sarkar and I met and what this has led me, how that has impacted my life. So we first met when I was an undergraduate student completing my biology degree at a small university called Barry University. And I came to interview at Rutgers, and Dr. Sarkar was one of the one of the persons that I interviewed with. So when in our short discussion, I learned three things very quickly: is that he valued productivity. He was a great scientist that valued productivity. He had two daughters, one that had already published before even entering university. And the third thing, based on the conversations that we had, is that I was not coming to Rutgers. and Dr. Sarkar had made it clear that productivity was going to be very important. So I got very surprised a week later when I received my acceptance letter for Rutgers, and this led to me being in Dr. Deepak Sarkar's lab and really learning how important the bidirectional relationship between stress and the innate immune system could lead to a variety of different diseases, including stress-associated diseases. So after completing my studies in the Sarkar lab, I moved on to my postdoctoral studies, and I am now a research scientist at Georgia Tech and with a joint position at Emory University, continuing some of those investigations that I had done initially in Dr. Sarkar's lab. So without the things that I've learned in his lab, which were the importance of independence and interdependence, the importance of grit and perseverance, the importance of salesmanship that I continue to see Dr. Sarkar accomplish through just putting out good research, but also initiating interest to people that may not be familiar with the field, with his genuine interest in the research. I've been able to continue in this field, and I am grateful for his everlasting support through the years. So congratulations again, Dr. Sarkar, and I wish you many, many more years of success.
Thank you, Dr. Franklin. Next up, we have Dr. Ali Al-Yashari. He's a lecturer at the Ministry of Higher Education and Scientific Research in Iraq.
Okay, thank you very much. First of all, I would like to congratulate Dr. Sarkar for receiving his prestigious award from one of the most prestigious academies, such as NJAS Academy of Science. And also, I would like to express my happiness and the pleasure to be among you on this special day and this special event. For me, I joined Dr. Sarkar Lab in 2014 to start my journey doing my PhD. The time I spent at Dr. Sarkar Lab was joyful, gorgeous, and we were as a one family, actually, because we were doing everything together. And the most beautiful thing, we had a calendar on the door of the office. We put the birthdays of every single one in the lab, working in the lab, and we celebrate every time the birthday of one of us. And also we really enjoy decoration when we have the New Year events. And that's the beautiful memories I have actually. At Dr. Sarkar Lab, we shared happiness, passion, and we shared condolence too. During my journey, I have learned a lot of things, such as doing a valuable science and helping others who are in need, in addition to experience the success and how to move forward. Every time when I was asking Dr. Sarkar to do more scientific experiments, he was immediately replying me, okay, Ali, go ahead and let's do it. So that's what I really liked. So I put a lot of notes here because I'm not good at sympathy things, okay? Actually, that was very inspiring and encouraging. After Rutgers and after I came back home, Dr. Sarkar keeps asking me every time about how is the situation there? How is your family members? How are you doing? So this is really making me feel very, very nice and good. I have some memories. I call it funny memories. At Dr. Sarkar, in addition to doing a good science, I have learned how to fix a car scratches, because Dr. Sarkar, every single week, he had scratches on his car. So I was fixing that. So I'm good in fixing car bodies now. In addition to that, I have another one funny memory. One time Dr. Sarkar went to India for treatment, I think for 1 month. So as soon he arrived in India, he asked us to do Skype meeting. So just one day, and then he were asking us, what is the new data you have? So we pretend there is something with the connection just to escape. So this is like, I just wanted to tell you, Dr. Sarkar, after like 4 years. And the other thing I have, what I stick with, Dr. Sarkar gave me some advices. So one of his advices, he told me, when you go back to your country, try to build a unique thing to be a founder of it. So what I did actually, I put it on the top of my targets when I came back to Iraq. And also I got a good position actually. I'm the Associate Dean for Scientific Affairs. So I kept reminding what Dr. Sarkar told me to do. So thankfully I got a grant with the US Embassy. There is a program called IREX, and we had a partnership with Columbia University. So this is what I'm doing right now. And we will establish the first SFC. It is the Stress Fighting Center in Iraq in collaboration and in partnership with Columbia University. And the other advice he gave me, he told me, Ali, science is not enough. You need to have and build a good social relationship with others to be successful. So again, and finally, I wish you all a happy and safe and healthy life. And I would like to congratulate Dr. Sarkar for this prestigious award. Ali Al-Yassari, Iraq. Thank you.
Thank you, Al-Yassari, Dr. Al-Yassari. Next up, we have Dr. Chang Quinn Zhang. Are you there?
Sorry, I was muted. Hi, how do I make myself like big? Okay. Hi, Dr. Sarkar, this is Chang Qing. First, I would like to congratulate Dr. Sarkar for this well-deserved award. I did my PhD study with Dr. Sarkar from 2007 to 2013, during which I learned so much from him, both in academic and in ways of thinking. I can still remember clearly that there was a cytotoxicity assay to test the effect of NK cells on cancer cells that I started by following a protocol I got as a piece of paper. And after failed a few times, I was going to give up and say, saying it won't work. Dr. Sarkar worked with me about the details and guided me to try again by finding the level of the background noise and titrate the cell numbers to figure out the number and ratio of cells to put in that will give a reasonable result. I wouldn't have succeeded in many experiments like this and had my name in 10 publications with Dr. Sarkar's lab without his support and guidance. So this kind of troubleshooting ability is also very important for my future career. I really admire Dr. Sarkar's continuous dedication to scientific research. I will always treasure the knowledge I learned from Dr. Sarkar and apply it to work when I need it. Also, I would like to thank Maria and Abby for organizing this event. It's fantastic to see Dr. Sarkar as well as my old lab mates again, and also to follow up with the most recent research findings. Hope everything goes well with everyone and hope we will be meeting with each other again in the future.
Thank you, Dr. Zhang. I'm sorry, I forgot to introduce your title. You're a project coordinator supervisor at Novogene Corporation.
Yeah, thanks.
Well, next we'll go to Dr. Wenbei Huang, who is a research scientist at the Institute of Neuroimmune Pharmacology at Seton Hall University.
Hello, everyone. I am working actually as a research assistant in Institute of Neuroimmune Pharmacology at Seton Hall University. And also I am a biology student in PhD program. And I have attended the annual Pioneers in Endocrinology workshop every year that is hosted by Dr. Sarkar and his endocrine research facility. This annual endocrinology workshop has actually attracted researchers from various institutes within New Jersey. And I want to say thank you to Dr. Sarkar for providing this opportunity to present my work and to network with the professors and fellows from other institutes in New Jersey. And thank you for providing the chance to get to know the New Jersey academic community that in this annual event. And I also want to say thank you to Dr. Sarkar for his nice and kind words and senior instructor. And congratulations to Dr. Sarkar for this New Jersey Academic Science in Technology Advancement of Science Award. Thank you. Stop sharing.
Abby, you're muted.
We're going to give Dr. Sarkar a few brief minutes of time to say something, and then we'll conclude. Please unmute.
Unmute yourself.
It has been a really exciting day for my life. I think I really am very, very happy to see so many of my old colleagues and friends and fellows. I really want to first express my deepest gratitude and appreciation to the NJAS committee and its president, Dr. Maria Agapito. It's really unbelievable that I was selected for the first time. And not only her, but also the committee who selected for this award. I also want to thank all the speakers for this presentation in the research areas I deeply love. So it's so good to hear all these old friends I know of and talking about all those exciting areas and new development. I also like to thank of all the guests as far as from India, Iraq, China, and all over US for attending this seminar. Some of the time like Jirong Chang who was from Shanghai. I think it's about 4 o'clock in the morning. So he attended most of the time, but then he fell in sleep, so he couldn't give his talk. This was really one of the major benefits to have a Zoom meeting, because without this Zoom meeting, we cannot do this. And it has also been so nice to see so many of my students and fellows, collaborators and friends, which brought back to so many fond memories. I have missed you all. And really it's good to see them back all and see them all succeeding. And this is an incredible feeling. I have attended hundreds of seminars and conferences and have received many awards. But this NJAS Inaugural Advancement Award seminar is a special one. And it's the very most meaningful one for me. Thank you, thank you, thank you so much for all attending and making this so memorable. I just want to give a short speech and thank you again for attending and making my life so memorable. Thank you.
All right. Thank you. I just want to give a big thanks to Dr. Maria Aguapito. You have done a fantastic job organizing this event, and we're going to leave you with some closing remarks.
Thank you. Thank you all for coming. Dr. Deepak Sarkar, it has been an honor to be part of your science family and to actually be able to host and help host this event today. We share so many years. I have been your PhD student, and through all that journey, I think, like many of my peers have said, you have been a main person to give us and teach us to keep that resilience in science. Science is not easy, specifically in careers, specifically in women. You know, there's always challenges, but you have taught us that with persistence and resilience, we must overcome any challenge that we come upon. The journey, like I said, as a scientist is not easy. It is full of many challenges and sleepless nights. And a day like today makes all those challenges priceless. Congratulations. Although today we are honoring Dr. Deepak Sarkar with the inaugural Advancement of Science Award from the New Jersey Academy of Science, there is a great need right now to address, just as we are all here and we are all in the science field. I would like to take this opportunity to highlight our role as science leaders in our community. Today, we are at a very difficult moment in the United States. Science is being challenged, and we need to enhance our efforts as real scientists to continue delivering true facts. According to the 2016 study from Tufts Institute for Democracy in Higher Education, STEM majors vote at lower rates than students in any other field of study. As science leaders, we must encourage our students, colleagues, and family members to advocate for science. The current science issues of concerns include the escalating climate crisis, social inequalities, exacerbating public health inequities, and real threats to science validation. We need to work hard to make these points clear as we move forward with the 2020 elections. We should advocate for science, collaborate with other organizations that advocate for science, and engage in leaders about how science concerns in order to change the present course in our country. We, as scientists, should come together during election 2020 to select the leaders that focus on issues that are prominent and important to scientists. On behalf of the New Jersey Academy of Science Board and the seminar organizers, I would like to express my deepest gratitude to all of you who have come together today from various regions of the country and the United States for your participation, which has enabled us to conduct a highly intellectual seminar. Since the Advancement of Science Awards seminar is a biannual event, we look forward to seeing you again in 2022 when we shall reconvene to discuss another topical theme. Thank you all for taking the time to patiently listen to what I had to say. I wish you all a blessed day. Have a great day. Thank you for coming.
Thanks, Maria.
Thank you, Maria.
Thank you.
Thanks, Maria.
And please
Congratulations.
vote.
Yes, please vote.
Thank you. Thank you, Abby. Thank you, Sophie.
Yeah, thank you.
Thank you, Maria. Thank you,
Nice
Abhi.
to see you, Dr. Sarkar.
Dr.
Thank
Sarkar.
you.
Congratulations, Dr.
Thank
Sarkar.
you. Thank
Thank
you.
you, Maria. Bye, Dr.
Thank
Sarkar.
you, Maria. Bye.
Deepak, congratulations.
Oh, thank you.
It
Thank
was
you so
wonderful.
much.
It was
Are
wonderful.
you there?
It was wonderful, and this is
Very
really,
nice organization.
really what you have done in your career. This is what you can see. Is really, I am proud of your achievement.
Thank you so much.
Great contribution to the field and to the young generation, and for the future. Congratulations.
Thank you so much. It really means it. I appreciate it.
Hey, Suli.
Hi. Good to see you.
Great job, Abby. You did. You run the session wonderfully. Very nice share of the
Very
session.
good. Yeah.
Thank you for inviting. It was a really great pleasure.
Bye, everybody.
Bye.
Is that Sherry?